So our first presenter is from Andelyn Biosciences. He is Matt Niloff, and he is the Chief Commercial Officer. He's going to be highlighting Andelyn's full-service cell and gene therapy CDMO capabilities in development, characterization, and production of viral vectors for gene therapy. He's also going to share the available capacity they have. Welcome, Matt.
Thank you, Barry. Thank you for that introduction. Good morning, good afternoon, good evening, depending where you're joining us from. Today, I will review Andelyn Biosciences capabilities and a little bit about our capacity. Speaking of which, we are having our grand opening today for our new corporate facility where we do clinical and commercial manufacturing. So a very exciting day for us here. I'll walk through a little bit about what Andelyn is about, our history, talk about our various facilities and capabilities, and a little bit about our AAV platform. So we are a cell and gene therapy contract and development a manufacturing organization. We have a very long history working with AAV. As many may be aware, we spun out several years ago from a Nationwide Children's Hospital, and I'll walk through a little bit about our history from there, showcasing our long-standing experience working with AAV. We have a partnership and strategic investment from Paul Corporation, now merged with Cytiva. So they are an investor, and we work very closely with them and a tremendous amount of support there, technically and with supply chain as well. We have 3 facilities, and I'll talk about those today. We are a little bit over 225 employees today, so growing quite rapidly. A tremendous amount of experience and expertise. Looking at our combined people leader service years, about 523 combined years of experience in GMP, commercial manufacturing, quality, and regulatory support, and about 197 of our leaders combined experience in cell and gene therapy related to process development, analytical development, and manufacturing. You can see with the graphic on the right, from our combined time at Nationwide and Andelyn, in over 20 years, we've supported over 75 INDs that have gone through approvals that stemmed from over 450 GMP clinical batches. And with all of our 3 facilities, we have about 250,000 square feet of space. So being a full-service CDMO, we offer concept to commercialization. Basically, we can start from DNA, and bring you all the way through a clinical and commercial manufacturing. We do plasmid manufacturing, vector design, plasmid cloning services, and cell line development. Our process development capabilities include doing the development, tech transfer, process optimization, full scale-up, and process characterization. Manufacturing, we have suspension, we have adherent capability. We do go up to 2,000 liters capabilities here at our corporate facility. And then we also have fill and finish capabilities as well with primary labeling and packaging. All of the analytical development and quality control to support manufacturing, regulatory support, and we do have several different partnership models, whether you are a biotech, pharma, or an academic or nonprofit. So our vision is pioneering solutions that turn hope into reality. Our mission is accelerating the development and manufacturing of innovative therapies to bring more treatments to more patients. Our name is quite inspirational to us. The name comes from 2 pioneering patients that were treated with gene therapies. Andrew was 8 years old at the time and received the first U.S. investigational gene therapy for Duchenne muscular dystrophy back in 2006. Evelyn was 8 weeks old and she received, at the time, experimental gene therapy for spinal muscular atrophy. In 2015, this molecule would go on to become Zolgensma. So when you combine Andrew and Evelyn, you get Andelyn. So a little bit about our history, going back several decades to our first work with AAV when we were Nationwide Children's Hospital in the lab of Dr. Phil Johnson, starting to work with AAV, and then doing first transient transfection back in 1994. I'm not going to go through every year here, but you can see it's been a slow and steady development of experience and expertise. Setting up a viral core, doing production eventually for external clients, and then in 2013 is when we first were producing Zolgensma, which later spun out to Avexis that was ultimately acquired by Novartis. We opened up a second facility in 2017, doubled our capacity. We also opened up a plasmid production facility in 2019. 2020 was when we formally created Andelyn and spun out. We had a 200-liter capacity at the time. Our strategic investment by Paul and Cytiva was in 2021. And last year was a very busy year for us, bringing online various capabilities with our suspension cell line fully developed. We launched the Andelyn Plasmid Center for GMP-grade plasmids, 500-liter suspension bioreactor capability. And then we also opened our development center for all of our non-GMP work. And then last year, we began producing in the Andelyn Corporate Center, the ACC, where we do all of our GMP manufacturing. And as I mentioned today, we're having our formal grand opening. And we do have 2,000-liter bioreactors here that are underway and available for manufacturing by our clients. And you can see a list at the bottom here of some of the molecules that we helped to develop that have since been acquired by biotech. So we do have 3 facilities: the Andelyn Corporate Center, as I mentioned, where we do our GMP manufacturing. It's our corporate headquarters. All of our QC labs are here and warehousing. We have the Andelyn Development Center, we call the ADC. This is for all of our non-GMP development activities. So basically all preclinical, we can do tox, and all of our PD and analytical development will occur at the ADC. And then the Andelyn Plasmid Center is where we produce non-GMP and GMP plasmids. Everything is within about 20 minutes within the Columbus, Ohio area. So the facility itself for manufacturing, it's 180,000 square feet, state-of-the-art facility, designed, for purpose, with the latest clean room technologies. We have about 16 GMP modular suites. So this includes upstream and downstream solution prep and filling. Our QC labs are here, warehouse, corporate headquarters space. As I mentioned, we are up to 2,000-liter bioreactors here. The clean rooms are modular, built with AES, continuous resonance flooring system that are mechanically coded into the walls. Flexible, design for multiple platforms. We can move adherent and suspension technologies in and out for clients as necessary. All of the manufacturing suites are grade C classified. We have dedicated air handlers for each manufacturing suite, so you get 100% fresh air supply in each suite. Unit directional flow of people, materials, product, and waste. And we do have N+1 redundancy built into all of our critical utilities, air handler fans, and we have generator backup as well. As mentioned, at this facility, we have our QC, QA, and regulatory affairs support. You can see, so it's a pretty large facility, and you can see we also have future expansion space as needed to basically duplicate our current capacity. As the market grows, we'll be able to support it. So our capacity, we consider it very nimble. As mentioned, we have adherent capability where we can do 30 or 60 hyperstack sublots for suspension processes. We have a 50-liter bioreactor. Today 50 and 200, quite active in terms of scale where gene therapy is at. More processes that are looking at more prevalent diseases need to scale to larger sizes, and so we have the STR 500 and STR 2000 available as well. This is a view of the Andelyn Development Center that we've been doing process development since last year. We have opened 20,000 square feet, and we have another 22,000 square feet available as needed to expand. We do adherent and suspension processes at these facilities, and we do have segregation with positive pressure rooms for the various modalities and viruses that we work with. The nice thing about being under one organization and very close is that it's very seamless transfer for our clients when we are doing development for them and then transferring them for clinical manufacturing to the ACC. It's only about 15 minutes apart. We have similar equipment for non-GMP and GMP. It's the same platform process and cell line from R&D all the way through GMP operation. Same materials demonstrated at scale equivalency of alternatives. We have the same analytical assays and established release assays for assessing CPPs and CQAs, and we are on the same quality system. So that drives very efficient tech transfer from our development facility. The Plasmid Center is located on the 3rd floor of the Abigail Wexner Research Institute. We do GMP plasma manufacturing there as well, 100% single-use flow path. It's about 1,600 square feet of R&D plasmid production space and 7,500 square feet of GMP production space. We have the 2 rooms for 50-liter single-use fermenter and 3 rooms with 200-liter fermenters, those of which we are looking to bring online early next year. And we do keep P helper plasmids off the shelf. Tremendous amount of in-house analytical capabilities, QC testing for various aspects of in-process testing, drug substance testing, drug product release testing, stability testing. We do all that here in addition to the common assays that you would see with viral vector production. I would say about 90% of our analytical capabilities are in-house today, and we do outsource some testing for appearance, AAV aggregation, residual host cell DNA, sterility, mycoplasma, and a few others. So we do have a platform that has really been proven over the very large number of both non-GMP and GMP production runs that we've done over several decades. And by having a platform, our clients can really expedite their timelines, and with that, you get some serious cost savings. So our platform is high yielding. We show consistent product quality, very plug and play, which allows you to reduce your upfront investment, time spent in doing development. We have a library of platform assays. This further drives down reduction in cost and time of doing analytical development activities. There is no cell license fee until you get to manufacturing. So this is we feel a big positive for our clients to come work with us with our cell line. And then there are no upfront costs or time spent on the master cell bank or working cell bank. So we offer program de-risking and a smooth CMC pathway, seamless tech transfer from preclinical to commercial, lots of process experience, good track record of reproducibility and operational efficiencies. We have scale down models to support manufacturing in our process development labs, and we do characterization and validation as well. And we're always incorporating the principles of quality by design and fostering process understanding in a very collaborative manner with our clients. So just again, a summary of our very well-established AAV platform. This operates both in suspension and adherent HEK293 cell lines. We have off-the-shelf helper plasmids, and then a full library of platform assays. And so we can support our customers from the GMP raw materials with the 3 plasmids, for example, for transfection. And then through production, media harvest, upstream purification, purification, fill and finish, and then all the way through quality control testing and QA release. So our platform approach, our philosophy is to basically have a robust platform suitable for all phases that provides consistent high yields, recoveries, purity, and quality across the scales. We've never had a rejection with all of our fully released batches, and so we ensure seamless transfer from preclinical to commercial manufacturing with flexibility built in to accommodate multiple serotypes within our platform. Just a vast bank of data that we leverage for the platform. You can see here in process development and research and talks, we can start from the Amber 250 M system, going through Flask to the STR50 all the way to the STR200, and then we can tech transfer that over to manufacturing, where we have 200, 500, and a 2,000 liter scale. Excuse me. So in summary, we have the AAV platform where we summarize really in 4 key pillars. One is expeditious, right? So we have optimized workflows and serotype agnostic processes, standard materials, batch records, and this saves months of time in development and allows you to speed up getting to the clinic. It drives down program costs with that time savings and extends financial runways for a lot of nonprofits or early stage biotechs, and this maximizes speed to the clinic in a very efficient manner. We do deliver high quality products and are very proud of that. So products manufactured at Andelyn have a very high quality attribute for safety, purity, potency, and yield. And this, of course, is measured extensively during production through purification, release, and stability testing as well. The platform is fully configurable, and so this drives down time, faster optimization, and efficiencies. So depending on the nuance of your process and the serotype, we have many different levers that we can pull for the various technologies and the different unit operations to get you to a robust and scalable platform in a timely manner and very efficiently. With this platform and all of the data, we do have a tremendous amount of predictability with performance that can be expected. So with that, we provide a tremendous amount of confidence in achieving that performance and manufacturing at very high quality and producing very high quality products. And so with that, thank you for your attention. Hopefully you have a good understanding now of where we came from and what our capabilities are. And we are here to help our clients drive their gene therapy programs forward.
Hey, thank you, Matt. Great presentation. And I always love hearing the Andelyn name story. That is really a nice part of this. We do have some questions. For the benefit of those who may have joined late, I think you started off by mentioning you have a grand opening today. Can you tell us about that again?
Absolutely. So we built a 180,000 square foot facility with just a tremendous amount of capacity to support the gene therapy, cell and gene therapy industry as it grows. And we started manufacturing in the fall of last year, but we are having our formal grand opening today. It's a very large event with all of our employees and families and local government officials and other guests. It's going to be quite an affair. Almost 500 people, a very large band, food, games for the children. So we're very excited to have that today and formally open the facility.
Okay, great. Congratulations.
Thank you.
Now, one of the facility schematics showed future expansion space. Any plans for that yet?
Our current capacity fully supports the needs of the industry today with our clients. We don't anticipate needing to build out the future space probably for several years. There's been a little bit of a funding crunch after COVID, as everybody is aware, but funding is expected to open up again. More of the programs, there's just a tremendous number of programs in preclinical. Right now, we're expecting those to move forward, needing to get to the clinic and being fully funded. And so with that, over the next few years, if we should need more capacity, then we have the ability to build that out. There are no plans today other than what we expect from the market in the coming years.
Yeah, great. You're positioned well. So here's a question. So this is a big, complex question. So somebody has an AAV program. I'm sure it depends on where they are starting, but everybody wants to know, how quickly can you do this and when can you start? Do you have some case study data to share with us on how you've been able to deliver that?
Sure, we have a lot of case study, and so we can definitely provide that timeline. So there are a number of different situations that a client is in when they come with us. Certainly, if we worked with them in the past and this is a resupply, we can get them going fairly quickly. Probably begin GMP manufacturing within 4 to 5 months. If we have a client that has their transgene, their gene of interest, then we would take that in and go through a number of activities, yield assessment, and do some analytical development, downstream optimization, whatnot. Work on the plasmid, both tox and GMP, and then do a tox production for them, and then GMP. That entire timeline, probably looking at about realistically 17 months. If they come with their plasmids ready to go, that'll shave a couple of months off. So probably looking at about 15 months or so. If they don't need tox, then we're probably down to about 10 months. And then as I mentioned, if it's a resupply, somewhere 5-6 months. And then any number of other timelines within that range, depending on the specific situation with a specific client.
Thanks, Matt. We're going to have to move on. There are a couple other questions we didn't get to from Robert and Scott, so I would point them out to you. So, Robert and Scott, thank you. And Matt will be following up with you some good questions on tech transfer and filling systems. So thank you very much for those. And Matt, enjoy the big grand opening celebration today.
Thank you very much, Perry. Take care, everyone.